In the third week of development, a human embryo is a flat disc made of 3 distinct cellular layers stacked together. Yet every single neuron, synapse, and processing center in the central nervous system will be constructed entirely from the simple, flat outermost layer of this disc, known as the ectoderm. The trigger for this construction comes from a layer down. Just beneath that outer surface lies the notochord, a rod-like structure that acts as the master conductor for early development. The notochord begins secreting specific proteins, sending an invisible chemical signal upward to the cells resting directly above it. The cells in the outer layer react physically to those chemical messengers. A specific strip running right down the middle of the back begins to multiply and swell, thickening into a dense band called the neural plate. This silent chemical signaling sets the entire sequence in motion, transitioning the embryo from a uniform cluster of cells into an organism with a defined neurological blueprint. Once the flat neural plate is established, the cells themselves stretch and elongate to prepare for a massive mechanical shift. The plate's center hinges downward, pulling tissue into a U-shaped trench called the neural groove. The outer edges are pushed into steep walls, or neural folds. These folds arch inward, fusing across the midline to form a sealed hollow tube. This moment is neurulation. The tube doesn't close all at once. Fusion starts near the embryo's middle and works outward, zipping shut simultaneously toward the head and tail. Through this precise folding process, a vulnerable patch of exposed surface tissue is successfully converted into a secure internal biological conduit. This zippering action happens with incredible speed. Sealing the entire length of the tube requires millions of cells to divide, migrate, and reorganize, demanding immense biological energy. To power that rapid division, the embryo relies on a specific nutrient, folate, or vitamin B9. Folate drives carbon metabolism, necessary to synthesize DNA. It supplies raw materials the cells need to multiply, fast enough to close the seam, before the next phase begins. Once the seam is sealed, the newly formed tube detaches from the surface layer, which will become the skin, and sinks inside the body. Meanwhile, cells left at the borders detach and migrate outward to form the peripheral nerves. While the genetic instructions provide the structural blueprint, the mechanical folding process cannot physically be completed without the exact nutritional fuel provided by folate. By the end of the 4th week of pregnancy, the neural tube is completely closed from end to end. Almost immediately, the tube's top swells, dividing into 3 primary brain vesicles. These spherical segments act as the foundational architecture for the brain, developing into the forebrain, midbrain, and hindbrain. Below them, the tube remains a straight drop, elongating to become the spinal cord. The precision of this early developmental window is critical. If the neural folds fail to zipper completely shut at the upper or lower ends of the tube, the resulting gaps cause permanent, often severe, neural tube defects. Because this folding happens so rapidly, often before a pregnancy is even confirmed, having sufficient folate already present in the body is the most effective way to ensure the tube has the fuel it needs to close without error. Over the span of just a few days, a flat sheet of cells folds inward, zips shut, and builds a tube. That brief, silent cellular dance lays the permanent physical architecture for all future human thought, movement, and experience.